Hello and welcome back to more human physiology. So let's finish off our discussion on membrane transport by having a short discussion on active transport. So before, we've talked about several different types of passive transport. We've talked about both simple diffusion and facilitated diffusion. While those two modes of transport are going to differ depending on whether the solute in question that's moving across the membrane requires a transporter of some kind, both of them in common kind of have the thing in common that neither one is going to require energy, because whichever solute you're talking about, whether it's simple diffusion or facilitated diffusion, the solute in question is moving down its concentration gradient. It's moving from an area of high concentration to an area of low concentration. As long as the transport is going downhill like that, it's never going to require any kind of energy expenditure. Now, active transport is a different story. So with active transport, we are going to be talking about moving a solute against its concentration gradient, or uphill if you prefer, meaning the solute is going to move from an area of already low concentration to an area of high concentration. So we're trying to make a region even more concentrated than it already was. So that idea constitutes one of the major types of active transport. We are pumping a solute from low to high concentration. The other mode of active transport is, as we kind of promised in a previous video, this is going to be how we deal with solutes that are just way too big to ever make it through the membrane, transporter or not. So this is going to deal with things like exocytosis, endocytosis, and other types of bulk transport. So let's start by talking about the first type of active transport, called primary active transport. And the prototypical example of this is something called the sodium-potassium pump, which we're going to use as a case study here. So we've already talked in Chapter 2 about how the intracellular fluid and the extracellular fluid are very different in terms of their solute composition, especially when you're talking about sodium and potassium. So if you will remember, they differ in sodium and potassium in the sense that the intracellular fluid has a very high concentration of potassium and a very low concentration of sodium. And that is flip-flopped when you're talking about the extracellular fluid, which has a very high concentration of sodium and a very low concentration of potassium. So in order to maintain these very steep concentration gradients, meaning there is a concentration gradient for sodium in which it wants to enter the cell, and there is a concentration gradient for potassium in which it wants to exit the cell, there will be times that we will see in the future in which the cell can open a sodium channel to let it into the cell. There will be times in which the cell can open a potassium channel to let it out of the cell for reasons that we will discuss soon. If we were to do this, we would dissipate these gradients. If we were to allow these sodium and potassium channels to be open all the time, we would eventually reach equilibrium, in which the concentration of sodium is the same inside and outside the cell. The concentration of potassium is the same inside and outside the cell. And we do not want that. We want to maintain this disequilibrium. We want to maintain the status quo in which there is a lot of potassium outside the cell and not much inside, and the same for sodium inside. So the way that we maintain this is this transporting pump called the sodium-potassium ATP-ACE pump. The ATP-ACE part of that indicates that this transporter is capable of hydrolyzing a molecule of ATP in order to extract the energy out of it. So the sodium-potassium pump utilizes the energy of ATP to pump both sodium and potassium against their respective concentration gradients. So if you'll notice in this picture here, if you start on the left and kind of move to the right here, the first thing the pump does is it binds to three sodium ions and expels them out into the extracellular fluid. So we're taking what little sodium is inside the cell, and it's not much, and we're expelling those out into the extracellular fluid. And we do that by simultaneously binding to a molecule of ATP and hydrolyzing it to give us the energy we need to pump sodium that way. At about the same time, we take 2 ions of potassium, which are in the extracellular fluid, and just like with sodium, there's not much potassium in the extracellular fluid, and we pump it the other way. So sodium is leaving the cell and potassium is coming in, and both of them are going uphill against their respective concentration gradients. So because we are going uphill in both cases, we need energy in order to get this done, and that energy comes from ATP. Now, the other method of utilizing energy in order to move a molecule against its concentration gradient comes from what is called secondary active transport. So primary active transport, which we saw in the case of the sodium-potassium pump, with primary active transport, your energy comes from ATP, and that's it. With secondary active transport, your energy is not really coming directly from ATP. It's actually coming from something else. So the best way to illustrate this is to look at a process as a whole. And I really love using this example because not only does it allow us to look at secondary active transport, it also allows us to see applications of both facilitated diffusion and primary active transport. So what we are talking about here is intestinal glucose absorption. So we've talked in the previous 2 chapters about the need for us as humans to eat food, digest that food, and then absorb it into the bloodstream so that we can deliver things like glucose and amino acids and fatty acids to cells that need those nutrients so those cells can utilize those nutrients to make ATP through cellular respiration. Now, when you eat food, you obviously swallow it. You start breaking it down in the stomach, and then once you have broken down those macromolecules like proteins and complex carbohydrates, once you have broken them down into their monomeric form like glucose and some amino acids, we need to absorb those nutrients across the lining of the intestine, meaning across a thin layer of cells in the intestine. So that is what you are looking at here. In yellow here are the cells of the intestine. So up here at the top where it says lumen of the intestine, that is where all the food and all the stomach and all the kind of acidy stuff is. Down here at the bottom where it says extracellular fluid, this is the interstitial fluid that is right on top of the blood plasma. So this is where we want to get those nutrients. The nutrients start up here. We want to get them down here. So if we are going to, say, absorb something like glucose, we need that glucose to pass through 2 different plasma membranes. We want it to pass through this top side, which is called the apical membrane of this cell. We want it to go across the apical membrane to get inside this intestinal cell. And then once it's inside the cell, we want it to pass through the membrane on the bottom, which is called the basolateral membrane. So there are a total of 2 different transport processes that we need to happen for glucose here. We need it to cross the apical membrane to get into the cell, and we need it to cross the basolateral membrane to leave the cell to get into the extracellular fluid so we can get it into the blood. So let's take a look at our situation here. If you start at the top, you will notice that glucose in the lumen, where all the food is, all the digested food, glucose is actually at quite a low concentration. No matter how sweet something is that you eat, the concentration of glucose in the lumen is going to be lower than what is inside the intestinal cell. So we can already see the challenge here. We are wanting to move glucose from an area of low concentration to an area of higher concentration inside the cell. So we are trying to push glucose uphill. Therefore, we need to expend some energy in order to get that done. We could do that with primary active transport, but this is where we're going to see secondary active transport work out for us. So this is what secondary active transport is all about. If you've already got a very steep concentration gradient for something, you can allow that solute to move down its concentration gradient, and when that solute moves down its concentration gradient, it's going to release some energy. So if you roll a ball downhill, it's going to release some of that pent-up potential energy. that potential energy that gets released can be utilized to simultaneously pump something against its concentration gradient. So that is what you're seeing here in bullet point number 1 here. Glucose is moving from low to high concentration, so that's uphill. At the same time that glucose moves through, sodium moves from high concentration in the extracellular fluid, which we've talked about before, to its lower concentration in the intracellular fluid. So we know already that the extracellular fluid has a lot of sodium and the intracellular fluid does not. So we have this concentration gradient for sodium. We allow sodium to enter the cell so that it may provide the energy we need to get glucose inside the cell. Really is quite ingenious. So at this point, utilizing this secondary active transporter, which is called the SGLT carrier, we can get glucose inside the cell. Now, at this point, we have basically accomplished half of our mission. We have gotten glucose to cross the apical membrane on top here, so it is now inside the cell. Now it's a matter of getting glucose to move through the cell and exit through the basolateral membrane. Well, this is a much easier task now because we are dealing with glucose at a high concentration inside the cell and a low concentration in the extracellular fluid down at the bottom here. So, therefore, we can just allow glucose to move through an open carrier, and it moves down below into the blood plasma by facilitated diffusion. So that's what's happening in bullet point number 2 here. So after bullet point number 1 and bullet point number 2, we have pretty much accomplished our mission. We have gotten glucose to cross 2 different plasma membranes, one through the apical membrane to get from the lumen of the intestine to the inside of the cell, and then number 2, we have gotten it to move through the cell, down through the bottom, through the basolateral membrane, and now it is inside the extracellular fluid, and we can get it to the blood easily from there. But that is not the end of the story, and let me explain why it is not. So let's go back to number 1 here. You will notice that in order to get glucose inside the cell, in order to get 1 molecule of glucose inside the cell, we had to move 1 sodium ion inside the cell. So obviously we're not just going to move 1 glucose molecule. We are going to move thousands upon thousands, if not millions, billions, etc. So every time we move a molecule of glucose inside the cell, we move a sodium ion inside the cell. Every time we do that, what do you think happens to that sodium concentration gradient? So every time we move sodium in, move sodium in, move sodium in, the concentration of sodium inside the cell goes up, and the concentration of sodium outside the cell goes down. We can continue to transport glucose like this, but we cannot do it indefinitely, at least as far as we've described things so far. So eventually, if we keep doing this, we're going to get to the point where the concentration of sodium is the same inside the cell as it is outside. And once we do that, we no longer have a concentration gradient for sodium, and at that point, we can no longer utilize secondary active transport to transport sodium across that apical membrane. So the way we're going to solve this problem is we are going to use the sodium-potassium pump down here at the bottom with bullet point number 3. This is what the sodium-potassium pump's job is. It is to always maintain strong concentration gradients for both sodium and potassium. And a big reason why we want to maintain those concentration gradients is because having them around allows us to utilize secondary active transport to do a lot of energy expenditure and to get a lot of things done in the cell. So when we talked about primary active transport on the previous slide, and I mentioned that basically we want to keep these sodium and potassium gradients because it's important. This is a big reason why it's important. We need these gradients to provide the energy we need in order to do things like absorb glucose and absorb amino acids. So like I was saying, this example not only allows us to learn all about secondary active transport, utilizing a sodium gradient or a potassium gradient to pump something else uphill, but it also allows us to see in action facilitated diffusion and primary active transport. And you'll notice the only type of transport that is not happening here is simple diffusion. So as long as we are kind of in that line of thinking, let's talk a little bit of terminology here. So if you go to the previous slide, you will notice a couple of different things happening. If you look at the SGLT carrier, glucose and sodium are moving in the same direction through the same transporter. With the sodium-potassium pump, sodium and potassium are moving through the same transporter but in opposite directions. And then with this glucose transporter, the facilitated transporter, glucose is the only thing moving through and it goes in one direction. So we actually have names for these types of carriers. In the case of glucose, or something like it, being the only thing that moves through the transporter, we call that a uniport carrier. So uni meaning one. If there's only one solute moving through a channel or a carrier at a time, we call it a uniporter. The SGLT carrier, in which two solutes are moving through the transporter at the same time and in the same direction, we call that a symport carrier. The prefix sym refers to on the same side. So because sodium and glucose are on the same side and they move through in the same direction, we call it symport. In the case of the sodium-potassium pump, that is an example of what is called an antiporter. Anti meaning on opposite sides. So in this case, sodium and potassium move in opposite directions through the transporter at the same time. So it is an antiporter. All right, so to finish off this video, let's talk a little bit about how we deal with solutes that are just way too big in order to make it through the membrane. So as we're saying, your best example for this is basically going to be big proteins, right? So a protein, regardless of whether there is a concentration gradient for it, it's too big to make it through the membrane, it's too big to make it through a channel or a carrier, so we need an alternative mechanism to deal with things like this. So when a very large molecule like a protein needs to leave the cell, it does so through a process called exocytosis. So exo means basically exit, so if a protein is leaving or exiting the cell, think exocytosis. So basically, when a protein is made inside the cell, so when we have finished synthesizing a protein, which we'll talk about towards the end of this chapter, the protein is going to be packaged into a very small kind of package called a secretory vesicle, which is made of the same material as the plasma membrane, and it will migrate to the surface of the actual plasma membrane, fuse with it, and that causes the release of the protein into the extracellular fluid. So in this regard, the protein or something like it never actually interacts with the hydrophobic core or any part of the membrane. We actually package it inside a membrane itself, and then we trigger its release by getting kind of creative here. So physiologically speaking, the best example of this is going to be when a cell in the body, say, makes something like a digestive enzyme. So those digestive enzymes are proteins made inside the cells of places like the pancreas. And those proteins, once they're made, need to be released into the extracellular fluid so that they can get into the gut. Another example would be the synthesis of peptide hormones like insulin, glucagon, and a lot of the pituitary hormones like oxytocin and things like that. So same deal. These are things that are synthesized inside of a cell and then secreted via exocytosis into the extracellular fluid. In the case of hormones, that extracellular fluid will be the blood. In this electron micrograph here, these very dark spots you see here are secretory vesicles that contain digestive enzymes. This is a look at the pancreas that you see here. So basically the opposite of exocytosis, in which exocytosis was all about a protein or something similar leaving the cell, if something is going to enter the cell, it does so by endocytosis. So think of it like this. If something is going to go into the cell, think endocytosis. So this is how we get bulk materials from the extracellular fluid to come inside the cell. So this can either happen specifically or non-specifically. If it is specifically, we call this process receptor-mediated endocytosis, in which the plasma membrane has a specific type of integral membrane protein that binds to a particular substance in the extracellular fluid, and this triggers a coated pit to form, and that eventually will form a vesicle inside the cell in which that particular molecule or solute is captured and internalized inside the cell. So this forms a transport vesicle, which can then migrate to wherever it is supposed to go inside the cell. Pinocytosis over here on the right, I'm kind of going out of order here. So pinocytosis, if we start here at the bottom, it's just like receptor-mediated endocytosis, except no specificity. So basically, the cell is kind of grabbing for whatever it can get out of the extracellular fluid. It doesn't care what it grabs. It basically just forms a internalized vesicle, and whatever kind of got trapped in there gets into the cell. It's kind of like setting up a big trap in the woods. You could have an idea of what you want to catch, but whatever gets trapped in there, that's kind of what you're going to get. Phagocytosis deals with the endocytosis of very large particles. We're talking even larger than, say, a protein. So most commonly with phagocytosis, we're talking about the cells of the immune system capturing things like bacteria and other types of pathogens, and then internalizing them so that they can be destroyed by particular hydrolytic enzymes inside the cell so that we can neutralize those potentially harmful pathogens. And then the last thing we'll mention here, just as kind of a clinical case study, I'm sure most of you have heard of a disease called cystic fibrosis. So now that we've talked a good deal about membrane transport, we can talk a little bit about the pathophysiology behind this disease. We can actually kind of understand it a bit. So cystic fibrosis is a genetic disease, meaning that a mutation in a particular gene in the DNA, in this case it is a gene called the CFTR gene, this mutation causes the gene, the protein product of the gene, which is a chloride transporter that pumps chloride ions into the alveolar fluid of the lungs. This protein with this mutation does not get made properly, meaning the cells of the lungs do not actually make this protein. What this protein ordinarily does, as I was saying, is it pumps chloride ions into the alveolar fluid, and this raises the osmolarity of the alveolar fluid, and this allows water to follow into the alveolar fluid, and this allows the fluidy secretions of the lungs to be nice and wet and not too thick. Well, if a person with this mutation does not make that chloride transporter, these secretions are going to not have very much water in them, and they're going to be very thick and very mucousy and very hard to clear. So you can definitely understand how someone with this disease is going to struggle to breathe. They basically have a very thick goo in their lungs. Normally, a person that does not have this mutation is able to dilute down that gook with lots of water because of the actions of this CFTR transporter. All right, so that is going to do it for this video. The last few subjects for us to tackle in this chapter that is kind of refreshing us on our cell biology, we're going to talk a little bit about eukaryotic cells, which is what we are made of as humans, and we're going to get just a very basic grasp on some of the major players inside the cell, meaning organelles like the nucleus and the mitochondria. So I will look forward to seeing you next time.